So let's discuss how we would do a mutagenesis screen for new auxotrophic mutations in a haploid fungus. So let's get some terminology on the board first. A screen is defined as looking for new mutants. Typically what happens in a mutagenesis screen is that a mutagen is applied, something that will increase the baseline mutation frequency. This makes it more probable that we'll find mutants that we might be interested in. So typically the way this is done is you have a wild-type organism, in our case a wild-type fungus. We expose it to some sort of mutagen, and then we examine the progeny of the individuals that were exposed to the mutagen, and we examine the progeny for mutants of interest. So let's think about how we might do a mutagenesis screen to look for new proline auxotropes. Let's take that example that we just discussed. Fungi that would be unable to synthesize their own proline, and thus can only survive if the media, the minimal media that we make, is supplemented with proline directly so that they can take in the proline directly from the media and as a result survive. So here's how a mutagenesis screen would be done. We would start with organisms that have been exposed to a mutagen. So we take our wild type, we'd expose it to a mutagen, and then we would plate these fungi on a plate that had minimal media, which I'll abbreviate as just MM, and we would supplement with proline. If you think about this, the genotypes that will grow on this plate, so let's put some colonies on the plate, the genotypes that can grow on this media would be prototropes, so wild-type organisms, wild-type for producing what they need to grow on minimal media. That will work as well. That will grow on this media. This media will also allow for auxotrophs to grow, but specifically we would have to say proline auxotrophs. So wild type can grow and proline auxotrophs can grow. Now, for example, anything else that we haven't supplemented, say something that's unable to make its own another amino acid, say glycine. A glycine auxotroph will not grow on this media. It would need minimal media supplemented with glycine in order to grow. So the only thing that's going to grow in this media are things that can make their own essential components from a carbon source and inorganic salts, so from the minimal media. And the one exception to that is anything that's a proline auxotroph will be able to grow because we've supplemented the media with proline. Now, the way to find the proline auxotrophs that we are interested in would be to grow these same colonies on minimal media alone. In this case, on straight minimal media, the proline auxotrophs will now not be able to grow. So if we sort of draw a little chart here and say, who can grow on what types of media, prototrophs will be able to grow just fine on minimal media. Proline auxotrophs can grow on supplemented media with proline, but they cannot grow on minimal media alone. And this other example that we mentioned, glycine auxotrophs, they wouldn't be able to grow on either. So the question is, how do we find in this mixture of two genotypes, prototrophs and proline auxotrophs, how do we find the rare proline auxotrophs that we're interested in? The way this is done is through something called replica plating. What replica plating does is it takes the same colonies in the same spatial organization, the same pattern, and reproduces them on a plate that has different media. The way that this is actually accomplished in the lab is to use something that looks like a stamp. It's basically like a handle with a stamper on it, and it's covered with velvet. Velvet is a cloth that has lots of little projections on it, and it can be sterilized in an autoclave such that it won't have anything growing on it. What you can then do is take this stamper with sterile velvet and press it onto this plate, which we would call the master plate. And then what you can do, what that basically does, the velvet picks up a little tiny bit of each colony, and these colonies, of course, are all clones of an individual organism that has replicated and made a little colony. What the replica plater then does is puts and inoculates a small number of individuals from each colony in the same pattern onto the minimal media plate. So this would be called a replica plate. What you would then do is you would examine the pattern. And we'll test my artistic ability here. So if that was the pattern that we got on the minimal media plate, the replica plate, we would then simply look and compare the two plates and look for something that's missing here but is growing here. So for example, I've drawn it such that this colony here is missing on the minimal media plate. This would then allow us to go back to the master plate and pick that colony and culture it as a separate organism, a separate clone, and that colony would represent a proline auxotroph that we're interested in. Let's suppose that through our mutagenesis screen we found one proline auxotroph that we're interested in. We're going to designate it simply as Pro1 minus, as proline auxotroph number 1, and the minus is just going to remind us that it's deficient in proline. So this is a proline auxotroph. Similarly, we could do any number of mutagenesis screens to look for other different auxotrophic mutations. So perhaps this is a methionine auxotroph, so we'll call it MET1 minus. Now what we might be interested in is saying, okay, we've got these two mutant auxotrophs. We're interested in the mapping relationship between the two loci. So we can use these as an example, as phenotypes that we can use as an example to show how mapping is done in haploids. Now, just like you've learned before, mutant lines are mutant in only one thing. So this Pro1 minus line would simultaneously have the wild type allele of the MET1 locus. Similarly, the MET1 minus line would have a wild type allele of the Pro1 locus. Okay, so here are these two haploid organisms. And again, remember, because these are haploid, they are N, they only have one allele of each locus. So it's not appropriate to start talking about, you know, two alleles of the Pro1 locus, two alleles of the MET1 locus. These lines, as you propagate them on a plate, are haploid. Now, the way that you would cross them together would be as follows. If we're interested in mapping, we're going to have to cross these two together. So I'll just erase this one and put it here. Okay, so if we cross these two together, what we're basically saying is that they fuse to make a transient diploid. So this transient diploid is now a diploid, and it's also a dihybrid. So now we can draw the loci together. Okay. This organism will then undergo meiosis. This is now a transient diploid. And just like any other diploid, like we've talked about, it's capable of undergoing meiosis, except now when it makes haploid products of meiosis, these are progeny as opposed to gametes, which is what we would see in a diploid. So we can sketch out the 4 different possible outcomes. Let's start with the 2 non-recombinant ones. Okay, so that was one of the inputs to the transient diploid. That's another one of the inputs to the transient diploid. So we'll start with those. So there's the one input there. Let's do the other one here. Then there are the two recombinants. One will now be the double mutant, and one will be wild type. This organism is a recombinant that now puts together the wild type allele from this one line with the wild type allele of the other locus from the other line. What's interesting here is that all three of these would be auxotrophic, whereas this one would be prototrophic. Now, the question would be, what would you see if you happen to plate these offspring? So you do this cross, you get the transient diploids, they undergo meiosis and make new progeny, and then you plate these progeny on different plates. What would you see? If you plated these on minimal media, so we take this whole mix and plate it on minimal media alone, the only genotype that's going to grow is the recombinant. So let's say you plated 1,000 spores. Okay. Oops. Okay. Fungi produce spores. Let's say we plated 1,000 spores on minimal media, but only 100 grew. Okay, so we observed 100. That would mean of these 1,000 progeny that we had 100 of these that were prototrophic. Now, because this is one of the recombinants, it means that we also have the same number of the other recombinant categories. So there would also be 100 here. But we simply don't see them because they can't grow on minimal media. As we've seen before, if you can determine the frequency of one of the outputs, you can infer the rest. So if there's 100 of these, and there's 100 of these, and these are the two recombinants, they will be equal in probability, equal in number. That would mean that the remaining 800 are found in these categories, and they will be even as well. So we would say that there are 400 of these each, which adds up to our 1,000. And that's our 1,000 spores that we plated on minimal media. The 100 that grow, again, are this genotype here, the Pro1 plus Met1 plus prototroph recombinant. And the total recombination frequency would be 100 plus 100 divided by 1,000, or if you work that through, it would come to 20 map units. So these two loci, should we want to draw a map, we would place them as 20 map units apart. And that's one example of how you can do recombination mapping in a haploid organism.